My name is Ling. I'm associate professor in the Department of Chemistry and Biochemistry at the University of Maryland. My research is focused on developing chemistry tools that allow us to understand neurobiology and the causes of neurodegenerative diseases. Unfortunately, there's no current cure for most brain diseases. For example, Alzheimer's disease, Parkinson's disease, and a lot of neurodevelopmental diseases, because we don't understand the brain enough, especially at the molecular level. So my lab is hoping to understand the molecular fundamentals underlying those diseases, especially what causes the disease and how can we better treat those diseases by developing mass spectrometry-based chemistry tools. Specifically, we use a cell called iPSC-derived neurons to model the human brain. So we can take skin cells from human patients, and then we can reprogram those cells back into stem cell, and then we can differentiate those stem cells into human neurons, which allow us to get live human neurons in a dish. We can extract all kinds of biomolecules from those cultured neurons, for example, proteins, lipids, amino acids, neurotransmitters, and we can analyze, identify, and quantify them using a chemistry instrument. We call it mass spectrometer. We can specifically tag proteins inside neurons and look at them at the fluorescence microscopy, and that helps us understand what happens inside live human neurons when diseases are happening. We're pushing the boundaries of scientific discoveries, and we're making new discoveries every day. We developed a website called Neuron Profile to share what we learned about proteins in human neurons so that the researchers around the world can access our data and to use it to better design the drugs to target human brain. With students and postdocs in my lab, we're really hoping to push the field forward and also to understand what causes neurodegeneration and how can we better treat patients and change people's lives.